Hello everyone, today I'm Bharity yakha of third semester Bharity roll number 21 and SO224 of Mathematics department. I'm here to present my topic blood test. So here I would like to highlight my contents, which includes introduction, a mere diagram, principle, theory, result, application, its future scope as well as its references. So let's start our introduction. Basically, a blood test is a lab analysis of things that may be found in your blood. You may have blood tests to keep track of how well you are managing a condition such as diabetes or a high cholesterol. You may also have them for routine checkups or when you are ill. Blood tests are very common. They are ordered by healthcare providers to find out how well organs such as your kidneys, liver, heart, or thyroid are working, help diagnose diseases such as cancer, diabetes, coronary, heart disease, and HIV or AIDS, find out if your medicine is working to make it better, diagnose bleeding or clotting, find out if your immune system is having problems fighting infections. Diagnose anemia such as iron deficiency anemia, pernicious anemia, aplastic anemia, or hemolytic anemia. Find variations in hemoglobin such as HS, C, or E, which are common in people of African, Mediterranean, or Southeast Asian background. Monitor chronic health conditions and diseases. Find health problems in their early stages. So these are the types of blood tests: blood chemistry test, blood enzyme test, blood test for heart disease risk. Blood tests can give your healthcare provider a lot of information. He or she can see if certain elements in your blood are in normal range, but in many cases, blood are in a normal cases. Blood tests are only part of the information your healthcare provider needs to make a diagnosis of a health condition. You may need to have some other types of tests as well if necessary. For preparing a blood test, most kinds of blood tests you don't need to prepare. These tests are to see what your blood is like under normal conditions. For some blood tests, you will have not eat for a certain amount of a time before the blood test. This usually means no eating or drinking anything after midnight before the test. These tests are often scheduled for early in the morning. Your healthcare provider will let you know if you need to fast before a blood test. So usually in procedure, in order to test your blood, a technician called phlebotomist will use a needle to take a sample of blood. Tell the technician if the sight of needle makes you nervous. He or she can help you feel more at ease. You can also look away during the procedure or bring a family member or friend to help distract you. After the procedure, your blood sample will be sent to a lab. Trained technicians then look for the information the healthcare provider has ordered. This may take a day or up to a week or more. Check back with your healthcare provider office to find out about the results. So this is how a blood is being taken for blood test. So its principle includes blood testing is the process of classifying blood based on the detection of antigens present on the surface of red blood cells. The correct determination of the blood type of donors and patients is crucial for the safe transfusion or transplantation of life-saving blood components, or transplanted organs and tissues. So blood tests are widely used to diagnose and monitor diseases, medication effectiveness, and organ function. Most analytics reside in the plasma, blood liquids component, and the separation of plasma from blood solid components typically entails sending blood samples to processing labs for centrifugation. But microfluidics offers the prospect of running tests on location at lower cost with mere droplets of blood fresh from a pricked finger, wicking in ordinary paper, or incorporating filters into labs on a chip. So here we can see the device used for our blood testing. Let's see how the technicians usually do our blood testing. The main purpose of conducting this experiment is to understand the basic concept of ABO blood group system and to know our blood group and type. Usually we need toothpicks, blood sample, alcohol swabs, lancet, clasp, slide, biohazard disposal container, monoclonal antibodies. All these equipments will be readily available in the blood test toolkit if available. So firstly, take a clean glass slide and draw 3 circles on it. Unpack the monoclonal antibodies kit. In the first circle, add anti-A, to the second circle add anti-B, and the third circle add anti-D with the help of a dropper. Keep the slide aside safely without disturbing. Now wipe off the ring finger with the alcohol swab and run gently near the fingertip, where the blood sample will be collected. Prick the ring fingertip with the lancet and wipe off the first blood of excuse me. As blood starts oozing out, allow it to fall on the 3 circles of the glass slide by gently pressing the fingertip. Now apply pressure on the site where it has picked and to stop blood flow, use a cotton ball if required. Mix the blood sample gently with the help of a toothpick and wait for a few minutes to observe the results. Here is a chart which predicts the different types of blood groups along its RH factor. These are the few precautions we need to consider before doing a blood test. Regarding the alcohol swabs, lancet, cotton balls and toothpick, we have to discard them. Now, drop all the materials including the glass slide into the biohazard disposal container after observing the result. As mentioned above, there are 4 major blood groups and 8 different blood types, collectively called the ABO group system. The groups are based on the presence of or absence of 2 specific antigens and antibodies A and B. Usually, Group A contains antigen A and antibody B. Group B contains antigen B and antibody A. Group AB contains antigen A and B both and no antibodies, and Group O contains no antigens and both A and B antibodies. So usually we have some questions regarding blood tests. So how long does it take to get a blood test results? This partly depends upon the urgency of the test. Most tests done at GP surgery are routine, meaning there is no urgency, so it may take a few days to get the results. Will I need other tests too? Sometimes the results of blood tests mean that the doctor may want to do other tests such as a scan on your heart or kidney or an electrocardiogram. I am scared of having a blood test. What can I do? Many people feel like this, and this is nothing to be ashamed of. Tell the person who is doing a blood test this will help them to look after you better. You could also ask someone to come up with you and distract you during the test. Some people also find breathing for relaxation helpful. If the problem is that you faint at the sight of blood, this is caused by the fail in blood pressure. You will learn techniques to stop this from happening. Some clinics can use a cream that numbs the area where the needle goes in. You should let them know in advance if this would help, as the cream may need to be applied at home in order to work properly. Unfortunately, there is no needle-free way of obtaining blood. So its result includes blood tests can help your doctor determine how different organs in your body are working. Examples of organs whose malfunctions can be visible in a blood test include your thyroid, liver, or kidneys. Your doctor can also use blood tests to search for markers of disease and health conditions such as diabetes. So, its application includes find out how well organs such as your kidneys, liver, heart, or thyroid are working, help diagnose diseases such as cancer, diabetes, coronary heart disease, and HIV or AIDS, find out if your medicine is working to make you better, find out if your immune system is having problems fighting infections, find health problems in their early stages. Usually its future score contains it can increasingly clear an extraordinary diverse range of clinically important conditions including infections, vaccinations, autoimmune diseases, transplants, transfusion reaction, and aging. The convergence of high throughput sequencing a growing role in clinical diagnosis. Example of high throughput sequence applications that have been begun to enter the clinic in recent years include cancer gene sequencing to identify clinically actionable mutations and whole genome, and metagenomic sequence to resolve medical mysteries. So lastly, I would like to highlight my references. Firstly, I draw the introduction from blood test, Johns Hopkins Medicine from the Google, blood test being taken, diagram from blood test, NHLDI, NIH from Google, principle, blood test typing, Sigma, Aldrich from Google, theory, blood test, Johns Hopkins Medicine from Google, results, https dot from Google, applications, blood test, Johns Hopkins Medicine from Google, future scope frontiers from Google.